that they have an outer membrane, which is mainly composed of lipopolysaccharides. So they also have this periplasmic space, which serves as the location of peptidoglycan synthesis. So dito po, for gram-negative organisms, hindi masyadong effective, if at all, against gram-negative organisms ang mga antibiotics such as penicillin. So this is because they have a lipopolysaccharide outer membrane that's covering their murine layer. Next are acid-fast cell walls. So acid-fast cell walls are structured pretty much the same as gram-positive cell walls, except that they have an extra layer on top composed of mycolic acid. And lastly, some bacteria, such as Mycoplasma and Ureaplasma, are unique in that they do not have cells. So what they have instead are sterols on their plasma membrane. Here at slide 8, we have another illustration comparing a gram-positive and a gram-negative cell wall. So again, their main difference is that gram-positive cell walls have a much thicker peptidoglycan layer, while the gram-negative cell walls have a thinner murine layer pero meron din silang extra polysaccharide outer membrane at the top. Okay, so upon microscopic examination, ito po yung magiging itsura ng mga gram-positive bacterial cells, which are purple, compared to gram-negative bacterial cells that turned out to be really dark pink or red. Next is bacterial morphology. Okay, so bacteria may vary in sizes and can occur in 3 basic shapes. So cocci, bacilli, and spirochetes. Cocci are round and spherical. Okay? So some of them may be arranged by twos. So yun po yung tinatawag natin na diplococci. Tetrads naman po kung they're arranged by 4, and if they are formed in a cube shape, and if they're together in a cube-like arrangement, then we call them sarcinae. Now, if the cocci are simply clustered together, ito po yung usual arrangement ng staphylococci and micrococci. And if the bacterial cells are arranged in a straight line, so resembling chains, this is most likely streptococci. Next, bacilli are rod-shaped, so mas elongated po yung itsura nila. So some bacilli may appear slightly rounded, and we refer to them as cocobacilli. So if the bacilli are arranged side by side, ito po yung tinatawag na palisades. And some bacilli, such as the Vibrio species, are known as the curved bacilli. So their cells look a little bit bent. Some bacilli may be spore-forming, so nagkakaroon po sila ng enlargement at different parts of the cell. So as we go through the semester, matututunan po natin sino specifically yung mga bacterial species that can produce spores. Next, as the name implies, spirochetes are spiral in shape. So they are coiled or curly. In addition to all of that, another terminology that we need to be familiar with is fusiforms. So kapag sinabi po na fusiform yung bacteria, this means that their ends are tapered, giving them a pointy appearance. And kapag sinabi naman po na pleomorphic, kapag pleomorphic po ang description ng isang bacteria, this means that they don't have a definite shape. So we could be looking at the same organism, pero yung ibang cells niya mukhang bacilli, tas yung iba mukhang cocci, or just sometimes they don't look anything like those 3 basic shapes. So ayan po, that's what we mean when we say pleomorphic. Alright, so before we get to the different staining techniques, ano nga po ba yung mga objectives of bacterial staining? So why do we do this? First is to determine the morphology of the bacteria, kasi po mas madali nating ma-visualize yung shape and arrangement ng mga bacterial cells if we stain them first before viewing them microscopically. Another would be to identify organisms with special structures. So aside from the fact that different bacteria have different shapes, some bacteria may also have special structures. So there are some bacterial stains out there that are specifically formulated for us to be able to better visualize these structures. So now, before we look into these special structures, let's first discuss what are the different staining techniques that can be performed in a microbiology laboratory. Alright, so first up is simple staining, which makes use of only one stain. So an example of this is methylene blue and also crystal violet, as shown here on these photos. Okay, so ayan po. Regardless of which organism is present in the sample, they're all the same color. Pero at least po, nakikita natin dito kung anong shape nila. So simple staining is only directed towards coloring the forms and shapes of cells. Next is differential staining. Alright, so differential staining, this is a technique or this is the technique that we usually perform in the laboratory. So this method aims to differentiate bacteria into separate groups. So for example, kung sila ba ay gram-positive or negative, or if they are acid-fast or non-acid-fast. And so dito po natin magagamit yung technique of differential staining. Alright, so there are four main steps in differential staining. First is applying the primary stain. Okay, so after we let that sit for a while and then wash it off, we then proceed to apply the mordant. So the mordant is not actually a stain, but it's a substance or reagent that is supposed to increase or strengthen the binding of the primary stain to the bacterial cells. And then we'll also let that soak for a while, then wash it off and proceed to applying the decolorizer. And then after washing off the decolorizing agent, finally we apply the secondary stain, more commonly known as the counter stain. So taking for example the process of gram staining. Ayan. So yung mga gram-positive organism po, or for example, if the organism is gram-positive, then they'll take up the color of the primary stain. Pero kung gram-negative yung bacteria dun sa sample natin, then even after applying the mordant, ayan, hindi pa rin po kakapit yung primary stain. So matatanggal siya nung decolorizer and it will now take up the color of the counter stain. So examples of differential staining are shown in these photos. Next is negative staining. So negative staining, this is a process where, or this is utilized to demonstrate the presence of diffused capsules that is surrounding the bacteria. So as we can see here in this example, pwede rin po itong gamitin sa mga fungi. So kaya po siya tinawag na negative staining is dahil baliktad po yung mangyayari. So imbis na yung mga cells yung pupulayan natin, yung background po yung magkakaroon ng kulay. So if there is a clear zone or halo surrounding the cells, then this confirms na meron nga pong capsule yung organism. So examples of staining technique that makes use of negative, or examples of stains that is used in negative staining is India ink or nigrosin. Alright, so hindi ko na po naisama dito, but there are also special stains that target specific structures of a bacterial cell. So in the example shown here, this is a flagellar stain. So this is used to better visualize the flagella of some bacteria, such as Proteus vulgaris. Alright, so here at slide 14, isa-isahin po natin yung mga surface polymers and cell appendages na pwede nating ma-observe after staining our samples. So first, a bacterial capsule is an organized material that is firmly attached to the cell wall. So capsules are made up of polysaccharide polymers, and these protect the bacteria by providing it with a virulence factor, which helps the bacteria avoid phagocytosis. Okay? Ayan. Maliban sa it helps avoid, or it helps the bacteria avoid phagocytosis. Tinutulungan niya din po ito to resist desiccation or drying out. So as we go through the semester, matututunan din po natin kung alin-alin yung mga bacteria na encapsulated. There's this other thing called the slime layer. So para din po siyang capsule, pero para din siyang capsule because they're both made of polysaccharides, but they are much loosely attached to the cell wall. And kaya niya rin pong tulungan yung bacteria to prevent phagocytosis. But aside from that, they also aid in the adherence of bacteria to the host tissues or to other materials such as synthetic implants or plastics. Onto cell appendages. Now, some bacteria may have flagella. So flagella are exterior protein filaments that can move, rotate, or turn, resulting in the bacteria to be motile. Okay, so may iba-iba rin pong arrangement ang mga flagella. Now, if the bacteria doesn't have a flagella, then we call it a trichus. If it has only one flagella, then we call it monotrichus. Now, if it has one flagella on each end, then we would call that amphitrichus. And if it has multiple flagella on one or both ends, then we would call them lopotrichus. Pero in some references po, kapag both ends merong multiple flagella, it may also be referred to as cephalotrichus. Pero ayun po, in general, we just simply use lopotrichus. And then kapag naman po, the bacterial cell is covered in flagella, so all over or all sides of the cell have a flagella, ayan po, we call it peritrichus. Next is pili or pilus, kung isa lang. These are non-motile, long, hollow protein tubes that can connect one bacteria to another bacterial cell for them to be able to exchange DNA. So wag pong kakalimutan yung term na conjugation pili kasi mamaya po magagamit ulit natin yan on our discussion on bacterial genetics. Okay, so again, the conjugation pili does not help with the motility, but it is used during transfer of genetic material. Next is fimbriae. These are proteinaceous hair-like appendages that are much smaller than pili and are responsible for adherence. So ito po yung size differences nila as shown here in this illustration. Okay, so kung kanina po, yung mga structures na napag-usapan natin, they can be found on the exterior of the bacterial cell. Let us now look into other structures that can be seen within the bacterial cytoplasm. Inclusion bodies are produced by some bacteria to serve as their energy source, food reserves. Okay? So just in case po mapunta sila sa isang environment na walang masyadong mapagkukunan ng nutrients. So inclusion bodies can also help lessen the osmotic pressure that may destroy bacterial cells. So listed here are examples of polyphosphate granules seen as inclusion bodies. So nag-iiba-iba din po yung tawag sa kanila depending on which organism would we find them in. So shown here on the photo to the left, this is the Babeson's granules or the metachromatic granules that are produced by Corynebacterium diphtheriae. Okay, so with the use of the Albert stain, ganito po yung itsura nila under microscopic examination. Next are endospores. So endospores are also known as asexual spores, and on the other hand, they are small, dormant structures inside of the cell that aids in the survival of the bacteria against external conditions. So kung yung inclusion bodies po kanina ay para kapag napunta yung bacteria sa isang lugar na walang source of nutrients, yung mga endospores naman po is para kapag napunta yung bacteria to places species that does not meet its preferred growth conditions. So endospores are composed of calcium dibicolinate, and they may be located either terminally, so sa pinakadulo ng bacilli, or subterminally, so sa medyo dulo lang ng bacilli, or centrally, so right in the middle of the bacilli. Okay, the photo on the left shows us a terminally located endospores that are produced by Clostridium tetani. Alright, let us proceed with microbial growth and nutrition. So ano-ano nga ba yung mga favorable environmental conditions ng mga bacteria for them to grow and proliferate? Okay, so for some bacteria, their source of energy may be light. So para silang mga halaman, no? They use light as an energy source, and ang tawag po natin sa kanila ay phototrophs. On the other hand, there are some bacteria that rely on chemical reactions known as oxidation. So kumukuha sila ng energy from the oxidation of either organic or inorganic compounds, and we call them chemotrophs. So same lang din po sila nung chemolithotrophs. Ayan. Those two terms may be used interchangeably. Now, for their carbon source, may mga bacteria po na carbon dioxide lang sapat na. Okay, so they use carbon dioxide as their sole source of carbon, and they are referred to as autotrophs. Now, some bacteria, on the other hand, they may need organic molecules from other bacteria, and we call them heterotrophs. All right, next is temperature requirement. So the optimum temperature for most pathogenic bacteria is at 35 to 37 degrees Celsius. Okay? So karamihan po ng mga bacteria na maaaral natin, 35 to 37 degrees Celsius po ang preferred incubation temperature nila. And they fall under the category of mesophiles. Now, there are some bacteria, though, that can grow best at 0 degrees Celsius. At para sa kanila, sobra na po yung 20 degrees Celsius. And we refer to them as psychrophiles or cryophiles. So, medyo maarte sila, no? Gusto nila sa malamig lang na environment. And an example of this is Listeria monocytogenes. So, Listeria monocytogenes is an example of a psychrophile. Meanwhile, meron din naman po mga bacteria that are thermophiles and hyperthermophiles. So they grow best between 50 to 125 degrees Celsius. An example of this is Thermus aquaticus. So the bacteria Thermus aquaticus, dito po nanggaling yung Taq polymerase na ginagamit sa PCR for DNA sequencing. So since they can stay active across a wide range of temperatures and they are able to withstand the process of protein denaturation during PCR. Ayan po. Next, oxygen requirement. So various bacteria may be referred to as either aerobes, anaerobes, or microaerophiles. So aerobes or obligate aerobes require oxygen for growth, and the incubation environment must have approximately 15 to 21% oxygen. For anaerobes, they are actually divided into 3 different types. So obligate anaerobes, facultative anaerobes, and aerotolerant. So obligate anaerobes will die after prolonged exposure to oxygen or air. So kapag nagko-collect po tayo ng mga samples that are suspected of harboring anaerobes, meron pong specialized transport and incubation system para sa mga organisms na to. Kasi otherwise, mamamatay sila imbis na mag-grow. Next are facultative anaerobes, so they can grow whether or not there is oxygen. But they would grow better in the presence of oxygen. Okay? Pero okay lang din naman kung wala. So those are the facultative anaerobes. For aerotolerant anaerobes, they can survive in the presence of oxygen, but they will be unable to process or they'll be unable to perform metabolic processes. Okay, so kung ikukumpara natin yung aerotolerant. Sa mga obligate anaerobes, yung mga obligate, they will die if there is oxygen in the environment. Pero yung mga aerotolerant anaerobes, as their name implies, they can tolerate oxygen. Yun nga lang, hindi nila kayang gumawa ng kahit ano. So they can't acquire nutrients, they can't multiply, they're just alive. But they can't do anything unless we actually move them to an environment with no more oxygen. Okay, so naiintindihan po ba? Those are aerotolerant anaerobes. Next are microaerophiles. Okay, so microaerophiles, again, as the name implies, they only require a small amount of oxygen, approximately 2 to 10% lang. So that is way below what aerobes need, which is at 15 to 21%. So again, microaerophiles require only 2 to 10% oxygen in the incubation environment. So an example of microaerophiles is the Campylobacter species, which requires only 5 to 6% oxygen. So now our question would be, ano nga ba ang meron sa mga aerobes at bakit po kaya bakit yung mga obligate anaerobes mamamatay sila agad in the presence of oxygen? So this is because aerobes are able to produce the enzymes catalase and superoxide dismutase. So catalase and superoxide dismutase are produced by obligate aerobes and also facultative anaerobes. And these enzymes are protective to the bacteria because they could counter the toxic effects of oxygen and its reactive oxygen species. All right, so yun po. Yung mga obligate anaerobes, wala kasi silang mga enzymes na to. So, hindi nila kaya mag-produce ng catalase or superoxide dismutase, which is why they can't survive long enough upon oxygen exposure. All right, let us proceed with those bacteria that require more carbon dioxide in the environment than usual. So capnophiles require a concentration of about 5 to 10% carbon dioxide for them to grow. And examples of these include Haemophilus influenzae, which mainly causes meningitis in children, and also Neisseria gonorrhoeae, which causes gonorrhea. Now, in this photo to the left, this is a candle jar that can be used to incubate both capnophilic and obligate anaerobes. Next is their pH requirement. Okay, for pH requirement, the optimum pH for most pathogenic bacteria is at 7 to 7.5. So karamihan po ng mga bacteria na maaaral natin, 7 hanggang 7.5 po yung pH ng mga environment that they choose to grow in. So neutral lang, okay? They fall under the category of neutrophiles. So if a bacteria would grow better in places with low pH, they are known as acidophiles. And if a bacteria would grow better in places with a much higher pH, then they are classified as alkalophiles. Other terminologies that we need to be familiar with are halophiles and fastidious organisms. So halophiles, they thrive best in an environment with increased salt concentration. An example po nito, yung mga Vibrio species. So karamihan sa kanila, they could be recovered or isolated from bodies of salt water. Kapag sinabi naman pong fastidious bacteria, they require a few other specific sources of nutrients such as additional amino acids or vitamins or heme. So generally, mahirap pong magpatubo ng mga fastidious bacteria because they don't usually grow using routine culture media. Let's proceed with the bacterial growth curve. Okay, so let's first familiarize ourselves with these terminologies para po mas madali nating maintindihan yung bacterial growth curve. Okay, so generation time of bacteria in a culture can be as quick as 20 minutes for fast-growing bacteria such as Escherichia coli, or as long as 24 hours for slow growers like Mycobacterium tuberculosis. Kaya sa mga microbiology laboratories po, usually, yung mga samples for bacteriology, nire-read po after 18 to 24 hours para po mabigyan ng time if, for example, is slow grower pala yung ina-isolate nila. Now, when we use the word generation, this refers to the doubling of cell number. So kung gaano man karaming bacterial cells ang meron sa sample mo initially, times 2. So yun po yung meaning ng generation. Now, when we say generation time, this is also referred to as the doubling time. So this is the amount of time required for bacteria to double their population. So kung gaano man katagal bago sila maging times 2 sa dami. Next, transverse binary fission simply refers to the most common form of cell division among bacterial cells. So, kaya po dumodoble yung population nila is because they undergo transverse binary fission. Okay, now that we know those terms, let's discuss the 4 stages of bacterial growth. So first is the lag phase. Okay, the lag phase or the period of rejuvenescence. Okay, so during this time, no cell division would occur, 0. Okay, so the bacteria is still adjusting into its new environment. Okay, so may adjustment period din pala yung mga bacteria. Now, while in the lag phase, the bacterial cells only begin biosynthesis, but there is still no change in their cell mass, cell size, or cell number. Second is the log phase or logarithmic phase or exponential phase. So this is now where the bacterial cells are actively growing and dividing. And ito rin po yung best time for us to collect samples for biochemical testing. So based on experience po, medyo hindi na rin po kasi accurate yung nagiging test results kapag luma na yung pinanggalingan na culture. Third is the stationary phase, also known as the plateau phase. So at the stationary phase, this is the period where there is a balance or equilibrium between cell division and cell death. So again, sa stationary phase, pantay na po yung dami ng mga bagong bacterial cells that are produced and those old cells that are dying. So during this time, metabolic activities of surviving cells are slowing down and the nutrients in the culture media are becoming limited kasi syempre nagamit na nung mga naunang cells na na-produce. Fourth is the death phase or decline phase. So this is where the bacterial growth and reproduction ceases and the number of dying cells surviving. So there's not much nutrients left, but there's so much toxic waste that has been formed, which further contributes to the death of the remaining cells. All right, here at slide 26, this is our visual representation of the 4 stages of bacterial growth, and we call this the bacterial growth curve. So the lag phase, adjustment period pa lang po. The exponential phase, the bacterial cells are now constantly doubling. So stationary phase, this is now the time where there is an equal number of cells being formed and those that are dying. And lastly, the death phase, this is now where no new cells are being created, not much nutrients is available, more cells are dying, and more toxic waste are accumulating. Okay, so again, this is the bacterial growth curve depicting the 4 stages of bacterial growth. Okay, so in this next segment of our discussion, focus tayo on the basics of respiration and fermentation. Alright, so respiration is an efficient ATP generating process wherein glucose is broken down. Ito rin po yung process na kayang gawin ng mga obligate aerobes and facultative anaerobes. So glycolysis, also known as the Embden-Meyerhoff-Parnas pathway, this is the first stage in carbohydrate metabolism, and this is the major route of glucose metabolism in most cells. So hindi lang po ito para sa mga bacteria. So during the process of glycolysis, glucose is oxidized to pyruvic acid. Next is the TCA cycle, or the tricarboxylic cycle, more commonly known as the Krebs cycle. So this is the most important process for the complete oxidation of a substrate under aerobic conditions. So definitely, hindi po ito kayang gawin ng mga obligate anaerobic organisms. So this enzyme system converts pyruvate, and then by the end of a very long and confusing process, energy is generated in the form of adenosine triphosphate, or ATP. So again, those are the processes involved in respiration, performed by obligate aerobes and facultative anaerobes. All right, so on to fermentation. These processes do not require oxygen. So ito po yung forms of bacterial metabolism na kayang gawin ng mga obligate anaerobic, facultative, and aerotolerant anaerobes. So medyo marami po ito, pero ang pinakailangan lang naman natin tandaan dito is kung ano-ano yung mga end products nila. First is alcoholic fermentation. So alcoholic fermentation utilizes glucose to produce ethanol. So ethanol is a form of alcohol, right? Maliban po sa mga anaerobic bacteria, kaya rin po itong gawin ng mga fungi and also protozoa. Homolactic fermentation, as the name implies, pyruvate is reduced to lactic acid and lactic acid only. So comparing this to the heterolactic fermentation, the end product of heterolactic fermentation includes lactic acid and also acetic acid and also formic acid, plus alcohol and carbon dioxide. Next is the mixed acid fermentation. So its end products include acetic, formic, lactic, and succinic acid, pero without alcohol or gas. So it's literally a mix of different acids. So malalaman po natin na mixed acid fermenter ang isang organism kapag nag-positive po ito sa methyl red test, which you'll learn more about sa laboratory classes niyo po. In butanediol fermentation, pyruvate is converted to acetone, and again, as the name implies, kasama rin sa mga end products niya ang 2,3-butanediol. So malalaman po natin na butanediol fermenter ang isang organism kapag nag-positive po ito sa Voges-Proskauer test. Take note though that medically significant bacteria can either be positive for methyl red or Voges-Proskauer, but never both. Okay po? So pwedeng mag-negative sila parehas sa M or at VP, but it can't happen that they're positive for both. Okay, naintindihan mo ba? So please don't forget that. Magagamit niyo po yung informasyon na yan pagdating nyo sa lab classes nyo. Well, actually, according to some online sources, there are a few serraceous species out there that can produce a positive result for both MR and VP. But for us, just as a general rule, the medically significant ones can't do that. Okay? Okay, so sana naintindihan po. Last one is the butyric acid fermentation. And as expected, kasama po sa mga end products yung butyric acid. On to the last segment of our discussion, let's proceed to bacterial genetics. Okay, so the bacterial chromosome is referred to as its genome. So this genome consists of one closed circular piece of DNA that is super coiled to fit inside of the cell, as we can see here in this photo to the left. Okay, so bali po, isang malaking loop lang yan ng DNA, but it is coiled very tightly para po magkasya siya sa loob ng bacterial cell. So now it looks like that. Maliban po dito sa pinaka-genome nila, bacteria also have what are referred to as plasmids. So plasmids are additional information contained into one small circular piece of extrachromosomal double-stranded DNA. So plasmids, they're not essential for bacterial growth. So okay lang po if either they don't have a plasmid or they lose it along the way. Pero po, genes that encode for things such as antibiotic resistance or toxin production or some other virulence factors, most of them are often located inside of those plasmids. Okay, so now, what are the different mechanisms of gene transfer among bacterial cells? All right, first is transformation. So transformation is the uptake of free DNA that is released into the environment when another bacterial cell dies, either by cell lysis or disintegration, which causes a rupture in the cell wall. Okay, so does anybody here play PUBG or any other games with the same rules as PUBG? Di ba po, kapag may player doon na namatay, you can loot their bag. You can take their things when they die. So parang ganun din po yung konsepto ng transformation. So when one bacterial cell dies and another bacterial cell stumbles upon it, they can take the DNA that has been released into its immediate surrounding. Now, after taking that free DNA, they can now incorporate this into their own genetic sequence by undergoing recombination. Next is transduction. So transduction is the transfer of bacterial genes by a bacteriophage. So a bacteriophage is a virus that can primarily infect bacteria. Okay, so again, bacteriophage are viruses that infect bacteria. So shown here is a colorized electron microscope image of a bacteriophage. Okay, so ang ginagawa po nito, it will infect one bacterial cell. And for example, this bacterial cell contains the gene for antimicrobial resistance. So pagkatapos niyang mag-replicate, chances are na-incorporate na niya sa sarili niyang genome yung genetic sequence that encodes for antimicrobial resistance. So now, the new copies of this bacteriophage will now move on to infect other bacterial cells that are nearby. And kung wala pa po sila nung gene that encodes for antimicrobial resistance, then this bacteriophage could pass it on to them as well. So that's what happens during transduction. Yung gene transfer is dala-dala nung bacteriophage and pinapasa niya to other cells that it's infecting. The third one is conjugation, also known as the horizontal gene transfer. So this happens when a living donor cell transfers its own genetic material to another bacterial cell with the use of its pilus. So remember, yung na-discuss natin kanina na conjugation pili. So ito po yung trabaho nun. It serves as a conjugative bridge to be able or to serve as a channel for DNA to be transferred. Okay, so literally it's using its conjugation pilus para iabot yung DNA niya dun sa susunod na bacterial cell. Okay, here at slide 33, we have illustrations on how each mechanisms of gene transfer would work. So first is transduction. So transduction, a bacteriophage infects a bacterial cell and passes on the genome that it has acquired from the previous bacterial cell that it has infected. Para naman po sa conjugation, a bacterial cell uses its conjugation pili to pass on its genetic material to another bacterial cell. And for transformation, and a living, competent bacterial cell takes up the free genetic material that has been released by a dead bacterial cell. Okay po, so here are the references that were used in the making of this discussion. Take note that our main reference was will be the textbook of Diagnostic Microbiology by Connie Mahon, Donald Leman, and George Manatix. So ito pong review handbook in Diagnostic Microbiology. This is actually a material used kapag nagre-review na po for boards. So I'm only using this as a guide para po maayos yung flow nung discussion natin, because I just think it makes more sense and it's easier to understand if our discussions are structured this way. Okay, so ayan po. That ends today's discussion. Thank you so much po for listening. Our chat box is now open for questions. So does anybody have any questions?